We will learn the first step of glycolysis, that is the phosphorylation of glucose. So, glucose in the presence of a hexokinase enzyme and coenzyme ATP converting into glucose 6-phosphate. This is a irreversible type of reaction and energy requiring step. The energy required here is obtained by the cleavage of a phosphoanhydride bond of ATP. Now, why this reaction is taking place? The first reason is that phosphorylated glycolytic intermediates cannot leave the cell as they lack transporters. We know the GLUT transporters are required to carry out sugar inside or outside the cell, but these transporters carry only non-phosphorylated sugar and not the phosphorylated sugar. So, the phosphorylated sugar remain in the cell, and without the use of any type of energy they remain in the cell. The second reason is that phosphorylate groups are essential components in the enzymatic conservation of metabolic energy. The energy which is released by the breakage of phosphoanhydride bond is conserved in the form of glucose 6-phosphate. The third reason is decreases activation energy and increases specificity. The activation energy of a reaction get decreased due to the binding of the phosphoryl group to the enzyme active site, and there is an increase in the specificity of the enzyme, because this is a best example of a induced fate hypothesis. That means the complementarity of the substrate with the enzyme get increased by the conformational changes in the enzyme structure. Now, what are the structural changes are taking place in this reaction? Here, the first structure is of a sugar, glucose. Now, ATP donates its phosphate group to the sugar. Now, where the change is taking place? At the position number 6, see, there is a hydroxyl group. The nucleophilic attack of phosphoryl group is taking place at this hydroxyl group in the presence of enzyme hexokinase, and this hydroxyl group is converting or changing into the phosphoryl group. Now see, the instead of hydroxyl group, here is the presence of a phosphoryl group. Now this is a best example of a nucleophilic attack of the phosphoryl group. The enzyme hexokinases carry out this reaction. Kinases are the types of transperases, the main class of a enzyme. Now the actual substrate is not ATP, but it is MgATP. Mg sits negative charge of the phosphoryl group, makes terminal phosphate an easier target for nucleophilic attack by OH of a glucose. Now this is a isoenzyme. That means hexokinase have 4 types of enzymes. Isoenzymes are the enzymes which have similar functions but are coded by different genes. So there are 4 types of hexokinases: hexokinase first, second, third, and fourth. The type 4th hexokinase have some different properties, and they are called as glucokinases. Thank you.